So this is where we got cut off in our last video, so we're going to pick up right here. This is going to be our part 2 of this unit, chapter 17 for Biology 102. So we were talking about leaves. Leaves represent the second component of the plant shoot system. They are considered the primary photosynthetic organ. This is where we're going to find all of the chloroplasts. Remember, the chloroplasts go through photosynthesis and they make the glucose and the sugars for the plant. There's going to be two types of meristem that's going to be found in leaves. We have plate meristem, and then we're going to have what's called marginal meristem. They're going to initiate leaf tissue development by coordinating cell division. They produce the main leaf structures, and they're also going to determine the point in which the cell proliferation switches to cell differentiation. So proliferation is where they're just going and dividing and making more or making that plant grow. Differentiation is where they turn into different parts and start doing different things within that cell. They do not maintain continual cell division after a certain period of time, so they'll quit proliferating and then just go into differentiation and just start performing whatever action the differentiated tissue performs. Now with gymnosperms, they're going to possess a thick cuticle, sunken stomata, and transfusion tissue that's going to be found around the vascular bundles to help assist nutrient transport here. And so looking here, we have a gymnosperm. These are going to be things like our evergreens and our pine trees. Looking at the picture, you can see how you have the vascular bundles with xylem and phloem, and then you're going to have transfusion tissue that's going to go around these vascular bundles. Of course, you have those sunken stomata there, and you have that outer cuticle. Angiosperms, with their leaves, are going to have an epidermis that's going to be protected by a thin layer of cuticle. It's going to have numerous stomata. Now, the stomata are going to be located at the bottom or lower portion of the epidermis. In some instances, it could contain things called trichomes. So in this picture, you're going to see the cuticle, which is covering the top. That's that waxy coating that's going to help to prevent loss of fluids and evaporation. Upper epidermis, you're going to see there, which is going to be the upper portion of the outer covering for protection. Underneath the upper epidermis, you're always going to have that palisade mesophyll cell. So these cells are going to be kind of a type of mesophyll that is where photosynthesis will occur. Going down, you will see your xylem and phloem running together, and then you'll have your spongy mesophyll cells, which kind of act like their name. They're going to be weird and spongy shaped. They're going to be lots of little openings throughout, and then those are always found with the lower epidermis. Now, you're not seeing the stomata or the guard cells in this lower epidermis, but that is where they would be found. Now, another thing you need to remember with angiosperms and gymnosperms. Think of gymnosperms as gym, as a male. Males don't like flowers. Gymnosperms do not have flowers. Angie, however, as a female, she would love flowers. So Angie loves flowers, Gym does not. Now, both of these plants have male and female parts, so don't get that confused. But that's just an easy kind of way to remember it. Angie loves flowers, Gym does not. So gymnosperms do not have flowers. All right, so going on in through the ground tissue, we're going to talk about the mesophyll. That mesophyll is going to consist of an upper layer that we just showed you, that palisade mesophyll. Those are tightly packed together. They're elongated sclerenchyma cells, which are going to have numerous chloroplasts. So that's where your photosynthesis is occurring. The lower layer, or the spongy mesophyll, is going to be irregularly shaped, like I just talked about, and it's made of sclerenchyma cells. These are going to be loosely arranged to create air spaces for gas exchange. Now remember, we're going to have oxygen that's being produced as a byproduct from photosynthesis that's going to be pushed out through the stomata, and it's going to be bringing in carbon dioxide that it will use for photosynthesis. So that's going to be our gas exchange. Vascular tissue that we see here is going to exist as bundles that are embedded within the ground tissue that form parallel veins in monocots or net-like veins in eudicots. These are going to be responsible for transporting your nutrients throughout the leaf. The leaves of seed-bearing plants are going to exhibit great diversity to help increase surface area that's going to help it with photosynthesis. So the more surface area you have, the more photosynthesis it can perform. With gymnosperms, they are going to be perennially evergreens, so these leaves are going to be green all year long. This will be things like ferns, strap-like, needles, scales, and fan shape. Angiosperms will exhibit leaves of different colors, shapes, textures, venations, structures, and arrangements. You're going to have two main structures of angiosperm leaves. You have what's called the petole. That's going to connect the leaf to the stem. And so you see this here in the picture. That's where it kind of divides off and makes the stem and then goes into the leaf. And you'll have the blade. The blade's going to be a wide, flattened portion of that leaf. With these angiosperm leaves, they are going to be classified as simple or compound. And also you can look at them as being pinnately or palmately formed. Simple is going to possess only a single blade, where a compound will consist of several leaflets that are arranged in pairs, pinnates. And they can be attached to a single point of origin, and that would make them palmate. And so you have those simple diagrams below. They can be classified as alternates, opposites, or whorled. If it's an alternate leaf, that's what you're going to see in the first picture below. That's going to be leaves that are arranged singly and alternate on nodes along the stem. If it's going to be opposite, that's what you see in the middle picture. Those where leaves are going to be arranged in two per node located directly across from each other. And then the last picture is going to be a whorled classification or a whorled description. This is where leaves are going to be arranged in three or more per node. Leaf abscissions are going to result in leaves falling from that tree, and it typically is going to be observed during cooler, shorter days, mostly in autumn. So now we're going to our modified leaf structures. They could have spines. Think of things like cactuses or cacti. These are going to be sharp projections that function as defense mechanisms against herbivory. That's animals or insects that might come along and want to eat that plant. Storage, things like onion bulbs. Those are thick, fleshy leaves that are going to store water and nutrients. Traps, like a Venus flytrap. Those are going to be modified leaves of carnivorous plants that are going to be used to capture and digest insects. And they do this because they need nitrogen and they're unable to get it from the soil like other plants. Tendrils, like in garden peas, are going to be modified leaves that are used by a plant to wrap itself around an object for support. Cotyledons are going to be seedlings. These are going to be your embryonic leaves, the first leaves to come out. Bracts are going to be like in poinsettias. They're brightly colored leaves that surround and provide protection for a developing flower. And so here's your examples of each of those. I'll give you a second to look at them. Now moving on to nutrition. Nutrition or nutrients is going to help to facilitate growth, development, reproduction, and survival of that plant. Nutritional requirements of seed-bearing plants is going to include things such as water, carbon dioxide, and minerals. Water is going to be the primary source of nutrition for plants. It provides hydrogen and oxygen to construct large organic molecules of glucose and to promote metabolic activities such as photosynthesis and cellular respiration. So remember, after it makes the sugars through photosynthesis, then it's going to have to break them down in order to make ATP to give it the plant, or to give that plant energy in order to perform its functions. Carbon dioxide is going to be a supplement in addition to oxygen atoms, but it provides the carbon that you're going to need for photosynthesis. So this carbon is going to help to build those glucose molecules. Remember, the glucose is C6H12O6. So it gets that carbon and oxygen from the carbon dioxide and breaks them apart, and it's able to use that in order to make the glucose. Minerals are going to be coming in through the roots from the ground. The first scientist to suggest the importance of minerals, things like nitrogen, to a plant was Nicholas Theodore de Saussure in 1804, and he's going to use hydroponics. Now, hydroponics is a technique of growing plants in nutrient-based solutions rather than soil, and this is actually becoming a very popular way to grow different types of vegetables and things. If you go to a store, sometimes you can actually find different types of lettuce that's available that has been grown hydroponically. And you can get this lettuce and it still shows the roots and everything on the bottom. And it's basically a live living plant that has some water with nutrients in it that you can put in your refrigerator and you can grow it, or it continues to live for about a month or so as you're eating on it. Now with these minerals, they're going to be acquired by plants to produce vital molecules. They also help to maintain osmotic balance and act as participants in metabolic reactions. They also form reproductive structures such as seeds that are going to store some of the minerals as phyton and a crystalline, which is going to be a crystalline salt. Plants are going to require some minerals more than others. This is going to be known as macronutrients, while other essential minerals are going to be required in lower amounts and are going to be called micronutrients. So some macronutrients that most plants need is going to be nitrogen, potassium, calcium, magnesium, phosphorus, and sulfur. Some micronutrients, that's the smaller amounts that they need, are going to be things such as chlorine, iron, boron, manganese, zinc, copper, and molybdenum. Now, nutrient acquisition is going to occur in different ways. First, you can have what's called the aquatic seed-bearing plants. These are going to acquire nutrients from their muddy substrates. Then you have epiphytic species. These are going to absorb nutrients from crown humus, or they can rely on mutualistic relationships with other organisms. You can see both of these in the pictures below. Parasitic plants are going to connect to another plant by a haustorium, and they can be partially hemiparasitic or full holioparasitic. That means it's relying upon its host for its nutrients completely. So with these you see here in the picture, we have parasitic plants such as mistletoe that acquire their nutrients by means of haustorium that are going to attach within the vascular tissue of its host plant. So it actually starts to grow into the plant, grows into its vascular tissue, and it's going to siphon that nutrients from that plant. The carnivorous plants are going to secure nitrogen and phosphorus by trapping and digesting insects and arachnids. And so as they break these insects and arachnids down, they're going to do it using an enzyme that they secrete, a liquid enzyme. And so after it traps that insect or arachnid, it's going to start to break them down, turns their insides basically into mush, and it's able to absorb those into the plant. Next, it will open the plant back up, and it will drop the carcass that it doesn't use. Now, with nutrient absorption, it typically occurs through the root system. So you have absorption of water. This is going to be absorbed through aquaporins that are going to be embedded within the cell membrane of the root hair cells, and this is going to occur through osmosis. Remember, that's the diffusion of water across the membrane. Movement of water from the soil is going to be dependent upon concentrations of dissolved minerals in the root cells. Water always follows solutes, so we need a lot of these dissolved minerals there in order for the water to come in. If the concentration of dissolved minerals is higher in the roots, then the water, like we said, is going to move from that soil into the cell because water always follows solutes. Absorption of minerals can occur with the help of some carrier proteins by facilitated diffusion and active transport. Remember, that does require ATP. Facilitated diffusion moves minerals from areas of high concentration to low, while active transport requires ATP energy to move minerals from low to high. You can have structures referred to as mycorrhizae. These are going to help provide better accessibility for nutrients by increasing absorptive surface areas in the roots. We have nitrogen-fixing bacteria that are going to help to facilitate the uptake of nitrogen by converting atmospheric nitrogen into usable nitrogenous molecules such as ammonium, nitrate, and nitrite. And so looking here, you can see that the mycorrhizae are going to be outgrowth extensions, where with the nitrogen-fixing bacteria, they're going to form root nodules that you see here on these roots. And so the bacteria live in there. They're able to take the atmospheric nitrogen that's going to be found in the soil, and they're going to be able to convert it into the usable forms that plant can use. Nutrient transport in water. So this is going to be explained by cohesion tension theory. In this process, it was first demonstrated by an English botanist, Stephen Hales, in 1727, with the concept of the theory proposed in the mid-1890s. He proposes that xylem sap is going to be transported from the roots to the leaves when transpiration from the leaves creates tension on water molecules. This is going to pull the chain of water molecules maintained by cohesion, that's where they stick to each other, and adhesion, where they're going to stick to the sides of the vessels, and so it's going to be able to use this cohesion and adhesion and pull it up through the xylem. And so this is kind of referred to sometimes as capillary action. Marcello Malfatti in 1686 is going to demonstrate that the physiological process of translocation through girdling is a process that removes rings of bark from around the trunk of a tree, displacing all tissue but the xylem. The experiment showed the accumulation of sugars collected above the girdle region, while the subsequent days to follow, the area below, as well as the roots, were going to be unable to receive these nutrients, and it's going to result in the tree's death. So water can come up, but nutrients cannot go down and feed the rest of the plant. With translocation, we have what's called bidirectional movement of phloem sap. That's going to be water that dissolves sugar, mainly sucrose, but may also contain some amino acids and hormones, enzymes, and a variety of other molecules. This is going to occur from a plant organ that produces, stores, and releases sugar, things like leaves or roots, and it's going to be known as a source to an area requiring and consuming sugar for growth and development, known as a sink. So sinks are going to be your roots, your flowers, or your fruits. This is going to be explained by what's known as the pressure flow theory. It was proposed by a German botanist, Ernst Münch, in 1930. This explains how sugar in phloem sap is going to be translocated by active transport from a source to a sink due to an increase in hydrostatic pressure within the phloem. Plant hormones are going to be very important. They're going to be a diverse group of chemical signals that are responsible for regulating growth and development and influencing a response to environmental stimuli. It was first detailed in a plant hormone provided by Charles and Francis Darwin in The Power of Movement in Plants in 1880. First experiments to demonstrate the existence of chemicals that promote growth in plants are going to be independently performed by Dutch botanist Fritz W. Wendt in 1926 and Russian botanist Nikolai Kolodandin in 1927. And so we see Darwin's 1880, where it shows that we have a complete stem that's coming up, and the shaded side is going to be where it starts to bend and it kind of grows towards the light. Then we're going to remove the tip, and that tip is going to be covered. With Wendt in 1926, it's going to show a complete stem. They're going to cut the tip off. They're going to use oxen-infused agar to help to grow it and unevenly apply that agar block to that plant, and then you're going to see how that plant is going to bend towards the light. So 5 main types of plant hormones. The first one is going to be called auxin. First plant hormone is going to be described by Charles and Francis Darwin, but it's identified and named by Fritz Kogel and Ari-Jan Hagen-Schmidt in 1931. It's responsible for promoting the elongation of roots and stems, initiating physiological tropism, that means growing towards things, and the differentiation of vascular tissues, stimulating structural organization of plant structures and inducing fruit development. Next, we have our second hormone, which is called the gibberellins. These are going to be named after a fungus that was defined in 1926 by a Japanese scientist, Uedi Kurosawa. Studies are going to show that this fungus produced excessive amounts of a chemical that was responsible for causing increased stem elongation and foolish seedling disease. It results in rice plants collapsing after reaching maturity. Since its discovery, multiple functions of gibberellins in plants are going to have been documented, including the elongation of roots and stems, initiating seed germination, and production of blooming flowers and development of fruit. The third type of hormone is going to be called a cytokinin. This is going to be kinetin and zeatin. These are going to be discovered in 1950s and 60s and were found to stimulate cell division in plants, so mitosis. Also, it's going to be responsible for influencing the development of plant organs and promoting early seed development and germination. It also induces leaf senescence. Fourth is going to be your abscisic acid. This is isolated and named in 1960. It's going to help to regulate environmental stresses by promoting seed dormancy and closing stomata to reduce transpiration. So let's say if we have a warm-up early in the spring, Then we have a nice cool down before it actually warms up. This is going to tell those seeds to go into dormancy. Also, it will close the stomata during very hot times or very windy times. The fifth one is called ethylene. This is first observed in the 19th century as a premature senescence and abscission of leaves that occurred on trees located near pipelines, supplying the illuminating gases to these street lamps that they used to have back then. The effect on plants is first going to be demonstrated by a Russian botanist, Dmitry Neljubov, in 1901. From 1901 to the early 1980s, investigative studies are going to look at this gas, and several scientists are going to reveal its effectiveness at a major plant hormone. It's going to help to promote seed germination, it helps to ripen fruit, and it helps with organ abscission and senescence. It's going to be used to help make things like bananas that you see here ripen early or ripen quickly. So lots of times they will pick fruits and pick bananas, things like that, when they are not ripe, but they will spray this hormone on the plants and then send them off, and they're going to start to ripen as they transport. Lots of times you can look at bananas in the store and you can see where it's actually ripening in spots. So you'll see yellow spots on it matched within the green. And that is where that hormone actually landed on that banana peel, and that's where it is ripening faster than the part that it didn't land on. So the production of the hormone is going to be typically in response to mechanical stress, drought, herbivory, or infection. Sensitivity of plants to stimuli such as light and gravity is first going to be explored in the mid-1600s, with the earliest scientific descriptions of plant sensitivity provided by an English author, Thomas Brown in 1658, and English botanist Robert Shureck in 1672. Plant sensitivity is going to continue to be studied throughout the 1800s, but growth of plants in response to stimuli, known as tropism, is going to begin with experiments conducted by Charles and Francis Darwin in 1880. Hormonal influences associated with tropism are going to later be documented by Fritz W. Wendt and Nikolai Kolandany in the mid-1920s. They're going to explain that by the Kolandany-Wendt hypothesis, that it suggests that the asymmetric accumulation of auxin in response for tropic behavior of plants towards light and gravity. So basically, plants are going to bend towards light. As they're bending towards light, what's happening is we're going to have auxins that are building up in that shaded side. Those auxins are going to cause the cells to become elongated and to start to be able to grow longer on the side that's shaded than the side that is facing the sun or facing the light. So therefore, it allows it to bend and get as much light as possible. Phototropism is a growth of a plant response to light. The response is going to be the result of redistribution of auxin to shaded sides of the plant, which initiates the elongation of the stem on that side, causing the shoot to curve in that direction. So that's what I just described. And you can see the auxin is going to be represented by these yellow dots in the stem below. That's going to cause it, like we said, to curve towards the light. Gene phototropin is going to detect blue light, and it's going to be identified as a mediator in phototropism. Heliotropism is going to be an alternate form of phototropism and coordinates the movement of the plant with the direction and position of the sun. So helio means sun. Photo simply means light. Gravitropism is going to be a growth response to a plant to gravity. Starch-Statolith's hypothesis is going to propose that growth response, and controlled by a stratolith, is gravitational sensors are going to be located in the cells of the stem and the root cap. These are going to settle in response to gravity. Asymmetric distribution of auxin is going to follow the movement of these statoliths and is going to trigger the directional growth in this response. So you see those auxins are all located on one side, and so it's going to cause them to curve up and grow towards away from the ground. Sorry, not towards. Thigmotropism is a response of a plant to touch. Most common example of this growth is going to be in tendril coiling. This occurs when the distribution of auxin to the side away from the point of contact is going to be stimulated, and it's going to cause either side of the tendril, making it coil around the object. And so working alongside auxin is also going to be ethylene, which is believed to aid in the growth and curvature of this tendril around the object. Another stimuli is going to be what we refer to as the biological clock. This is going to allow plants to anticipate and match their physiological response to specific environmental cues. It's going to be responsible for maintaining different circadian rhythms to ensure growth, development, and metabolic activities. Circadian rhythms were first documented by Jacques de Morin in the 1700s, the investigations of Augustin de Candolle in 1832, and believed to have provided the first experimental evidence to support the idea occurring in plants. Clock genes, which initiate the expression of certain genes, are going to be responsible for stimulating circadian rhythms such as leaf movement, photosynthesis, and even seed germination. Photoperiodism is going to be a physiological response of plants to day length. The idea first is going to be proposed in the early 1900s, and by 1920s, Whitman Garner and Harry Allard are going to demonstrate flowering was going to be induced by photoperiodism. There's 3 types of plants that are going to be classified in accordance with their flowering response and their exposure to light. We have the first, which is called a short day plant. This is going to flower when exposed to shorter light periods. Second, we have what's called a long day plant. These flower when they're exposed to longer light periods. And then we have a day neutral plant, which flowers regardless of light periods. So with, let's say, the short day plants, these are going to flower the best in spring and in fall. The long day plants, of course, are going to flower the best through summer. And then the day neutral plants, it does not matter, they will flower regardless. Protein phytochrome was going to be discovered in the 1950s and be the key regulator in inducing the photoperiodic responses of plants. 2 types of photochrome proteins are going to initiate the response based on the absorption of red and far red light. During the day, photochrome PR is going to absorb red light and convert it to its alternate form, photochrome PFR. This is going to induce seed germination. It's going to inhibit stem elongation and set the internal clock and promote flowering. At night, the photochrome PFR is going to convert back to photochrome PR in the presence of the far red light. Plants are going to be under constant attack by pathogens and herbivores. Therefore, they have to evolve specialized defense mechanisms to help provide them with extra protection. Mechanical defenses are going to be your first of 4 types of defense mechanisms. These will include things like morphological and physical structures that offer the first line of defense against an attack, such as epidermis modifications like thorns and spines. Plants also employ thigmotropism in response to a reduced surface area that's available to a herbivore. Mimicry is going to be another plant defense that they use. It's evolved adaptation that allows a plant to imitate another organism to provide protection. So for example, we have the Passiflora plant, which is going to produce leaves that appear to possess eggs to discourage the Heliconius butterfly from laying its eggs on the leaves. If it were to lay its eggs on the leaves, then as those eggs start to develop, they will eat away at the leaf for a food source. Mutualism is where it's going to enlist an organism for protection while compensating it with food, housing, or both in exchange for its service. Examples of this, a relationship between the Cassia henneti trees, and we're going to have these ants. The ants are going to protect the tree from herbivores and pathogens, while the tree provides the ants with food and a house. Chemical defenses are going to involve using a secondary metabolite. That's a diverse molecule that's going to be stored in plant cell vacuoles. Several classes of secondary metabolites are going to be terpenoids, phenolics, cardiac glycosides, alkaloids, and cyanogenic glycosides. These are going to be known for their antimicrobial properties, their bitter taste, and of course their toxicity. They can interfere with nucleic acid synthesis, So that's the making of nucleic acids like DNA and RNA. They can interfere with neurotransmitters, digestive processes, cellular respiration, and sodium-potassium pumps. Some herbivores have evolved tolerance to plant secondary metabolites, such as the monarch butterfly. Monarch caterpillars are going to consume milkweed leaves that contain a cardiac glycoside, which is going to accumulate and become a defense mechanism against predators when the caterpillar becomes an adult. So it basically makes it poisonous. Benefits, ecological importances of plants. To maintain survival and biodiversity of other species by providing homes and reproductive areas within or on their roots and shoots. They're going to enhance the quality and availability of soil nutrients upon decomposition in their vegetative structures. So they also help to supplement nutritional needs of many animals through consumption of seeds, fruits, and even nectars. Flowering plants are going to rely on insects such as bees, butterflies, and moths, birds, hummingbirds, and small mammals such as bats for pollination. They maintain the integrity of the soil by preventing erosion, and they reduce carbon dioxide by sequestering the greenhouse gas within vegetative structures, and they're going to use that to perform photosynthesis and create glucose out of it. Human importance of plants. Manufacturing and household items such as baskets and pillows. We're going to use them in traditional ceremonial events and the production of jewelry, personal care products, leather, and rubber. Commercially important in providing lumber for construction of houses, while other cultures prefer to use different grasses or palm leaves to build their homes. Paper is going to be made from seed-bearing plants such as pine and birch, and the textile industry is going to produce clothing using cotton and hemp. They're going to provide a variety of foods, including sago and pine nuts from gymnosperms, along with different edible foods that are going to be supplied by angiosperms such as wheats, nuts, vegetables, and fruits, which are also going to supplement nutritional value in the form of vitamins and minerals. Many foods and beverages are going to be consumed by humans that are going to be edible and desirable because of their secondary metabolites that are produced by plants, including things like spices and, of course, my favorite, caffeine. Plants are going to have many medicinal importances. We have traditional herbal medicines, things like ginkgo, pine needles, and bark. We have ephedra that's going to be used in traditional remedies to relieve many respiratory ailments. So ephedra is known as pseudoephedra that we use in things like cough medications. They're also going to help things such as asthma, bronchitis, and symptoms of the common cold. Production of modern-day pharmaceuticals has become more familiar in the treatment of different ailments. So we have the pseudoephedrine, that secondary metabolite that's going to be found in ephedra to alleviate nasal congestion. Salicylic acid, which is going to come from willows to produce aspirin. Quinine, which is going to be from the Ciconia trees to treat malaria. Morphine from Papaver seminiferin to treat pain. Digitoxins are going to help to treat cardiac cancers, and then we have many anti-cancer drugs that have also been developed from seed-bearing plants. And so this is going to help to treat cancers that are ranging from ovarian cancer to leukemia. Other types of plants are being investigated right now for anti-cancer properties, including some of the two that you see below. And to finish us out, we have our horticultural importance. Seed-bearing plants are going to have adorned botanical gardens and landscapes since the ancient Egyptians and Assyrians. Today, this practice continues in parks and residential areas around the world, with many that are going to have planted ornamental plants, while others are going to grow for the sole purpose of providing food for their family. Nearly 50% of these plants are going to be purchased from home centers, with households spending around $350 for lawn maintenance and gardening activities each year. Now, I know I go to a lot of different garden places, garden centers and home centers, and I buy a lot of plants. I buy a lot of herbs for spices and for cooking, but I also buy a lot of ornamental plants because I just love to grow these, mainly angiosperms. Okay, so that's going to finish us up for this unit. We're actually going to come back. That's going to finish us up for plants totally. We're going to come back and we will start our unit on animals next.